Hello, everyone, and welcome to our video brunch. We're going to talk about new therapies and new approaches to treatment in IBD. I'm Dr. Aline Charabaty, founder of Monday Night IBD and Associate Professor of Medicine at Johns Hopkins School of Medicine and the Clinical Director of the IBD Center at Hopkins Sibley Memorial Hospital in Washington, D.C. I'm joined today by my colleague and very good friend, Dr. Bruce Sands. Dr. Sands is the Dr. Beryl Krohn Professor of Medicine at the Icahn School of Medicine at Mount Sinai and Assistant Chief of the Division of Gastroenterology at Mount Sinai Health System in New York. Bruce, welcome and thank you for joining me.
Thanks for having me, Aline. Nice to see you.
Our video brunch series is CME certified and jointly provided by Global Education Group and bonumce. These activities are supported by education grants from Janssen, AbbVie, and Takeda. Our disclosures are shown on screen. If you haven't already completed our pre-activity survey, please do so by clicking the link below this video or by scanning the QR code shown here. Let us know who you are in the polling questions below. And please make sure to complete your evaluation at the end of the session so you can claim your CME credits. And of course, follow Monday Night IBD and bonumce and set your notification so you can be aware of new CME certified programs. Upon completion of this educational activity, participants should be able to evaluate the clinical efficacy of new therapies, emerging therapies, and combination therapies. So we covered some of the new therapies coming in our IBD toolbox, but I want to talk now about IL-23. And these are therapies that have been shown also to be very effective and safe in treating Crohn's and UC. We have 3 anti-IL-23 currently approved for both UC and Crohn's, risankizumab, guselkumab, and mirikizumab. All these drugs have been initially approved for an IV induction followed by sub-Q maintenance, but now it seems like there's an evolution in the IL-23. The story didn't end there. So Bruce, can you walk us through the most up-to-date data that we have for anti-IL-23?
Yeah, sure. I think the new class of the anti-IL-23s, of course, a little bit different from ustekinumab, which blocks both IL-12 and 23. And you'll have to ask one of my immunologist friends, why does blocking one cytokine, why is that better than blocking 2 cytokines? I don't know why, but the Galaxy 2 and 3 studies clearly show that better than any other study because this was quite an interesting study design. It was only recently published in The Lancet. What was remarkable about this study is, number one, it had both a placebo comparator and an active comparator, which is ustekinumab, which we know is effective in Crohn's disease. Number two, this is a treat-through design study. So what you see is what you get at the end of week 48. It counts for every patient who came into the study. There was no re-randomization of responders. All the blinding was maintained throughout. So in many respects, this is the gold standard for studies, I would say. It was an IV induction regimen, 200 milligrams IV every 4 weeks, 3 doses, and then subcutaneous maintenance with 2 different regimens, either a less intensive regimen of 100 milligrams sub-Q every 8 weeks or a more intensive regimen of 200 milligrams sub-Q every 4 weeks. And regardless of the regimen, you can see that both of those maintenance regimens with that IV induction were certainly effective. I'm showing you endoscopic response because it's really an objective response here, but you can understand that the clinical remission rates were the same and the study achieved basically all of its proposed outcomes. You can see that regardless of which dose they used, first of all, high rates of endoscopic response at week 48, roughly 50% across the board, a little bit higher for bio-naive, but still quite high for the bio-inadequate responders. But also look at the difference between guselkumab and ustekinumab. It's somewhere between 10 and in some cases 15% difference, depending on whether it's bio-naive or bio-inadequate responders. And, we've approved drugs in comparison to placebo based on 15% superiority, but this is to an active comparator that we know is effective. So I find that very remarkable. In addition, we also had the publication recently of the Gravity study, which in many ways is a replication of what we saw in Galaxy, except here we're talking about subcutaneous induction, 400 milligrams subcutaneously every 4 weeks times 3, and the same kind of maintenance regimen subcutaneously that I mentioned for Galaxy. And these results you could overlay on what I showed you for Galaxy almost exactly, showing you very, very similar results. So now for the first time, we have the ability to give subcutaneous induction and subcutaneous maintenance with a choice of more intensive or less intensive maintenance therapies to go for over a year. We also have evidence that you can perform subcutaneous induction in ulcerative colitis. This comes in the ASTRO study. Remember that we had the QUASAR study, which showed us that IV induction with guselkumab in ulcerative colitis was effective and maintenance as well. Well, here we're looking at the same thing, subcutaneous induction and subcutaneous maintenance. And guess what? It works too. But I think even more exciting for the future is the idea that maybe we can inhibit IL-23 without a biologic. So icotrokinra, a very difficult name, but I think we're all going to have to get used to it because these phase 2b data from the Anthem UC study were really remarkable. And these haven't been reported yet in full, but you can see here that the results for icotrokinra versus placebo were remarkably more effective. And so this is an oral anti-IL-23 peptide, and it does seem to work in ulcerative colitis. So we'll have to see how this plays out in phase 3 for sure.
All right, so we talked about new MOAs. We talked about MOAs that we're already familiar with, but with new method of delivery. Bruce, let's talk now about combining effective therapy or advanced therapy and what we're trying to achieve with that, either breaking the glass ceiling or finding combination therapy where combining higher efficacy and better safety. So tell us more about what's going on in this space.
Yeah, it's a very interesting space. We all realize that we're reaching a plateauing of efficacy with single individual therapies. So we're all looking for ways to break through that ceiling of efficacy. I think there are two ways forward, potentially, at least two ways. Well, maybe three. One would be invent an absolutely incredibly effective medication, but we haven't seen that yet. But more likely, another way would be precision medicine. We identify a predictive biomarker that tells us about a subpopulation that is going to have, let's say, an 80% remission rate to one particular mechanism of drug, but it's a subset of all patients. And the third way that I think is maybe most interesting and relevant nowadays is combining two effective therapies and seeing if you can produce more by combining them. And if you think about it, we've done this for a long time in IBD. We have learned from Sonic that if we combined thiopurines with anti-TNF, we actually get better outcomes than with either one alone. So this is not a foreign concept. But now we're seeing this applied to biologic therapies and potentially to small molecules with biologics. And there are a number of quite interesting studies that are going on. And the one that really set the field on fire in this direction is the VEGA study. VEGA intentionally combined guselkumab, which is an anti-IL-23, with golimumab, which is an anti-TNF, and looked at that comparison in the same patient and either one alone as monotherapy. And these were patients with moderate to severe UC. They were naive to either mechanism of action, it's important to point out. And this is an early phase study, a 2A study, just to see what you get. Now, if you look at the results, you can see that the primary endpoint of clinical response maybe doesn't look that exciting. You don't really see any remarkable differences between combination therapy or the monotherapies. But if you look at the secondary endpoints, where you have the much more stringent endpoint of clinical remission, here you see a near doubling of the efficacy of either monotherapy alone. So this really set the world on fire because it showed for the first time that combining two completely different mechanisms of action of drugs might give you something very, very different, a much better efficacy. And by the way, the safety also looked good. It did not kind of catapult the safety concerns forward, which is great. So on the basis of these results, we see the undertaking of more progressive studies, one in Crohn's disease called DUET-CD and the other called DUET-UC, obviously one for Crohn's and one for ulcerative colitis, looking at exactly this combination therapy. But here, we're also looking in patients who might have been refractory to one or the other mechanism of action. So that's a slightly different question. In other words, when you combine drugs, can you even overcome the lack of efficacy to one of the combination? So I think this is going to be a very important set of studies. These are difficult studies to do and quite large, but what you can expect is the outcomes to be released sometime in the coming year, perhaps. And this will inform the field quite a bit. If we see positive studies here, you can expect all kinds of studies of combination therapy. If these are negative studies, then I do wonder if people will be less enthusiastic about combination therapy in the future. There are lots of other studies coming down the pike that are attempting to look at dual targeted therapies or advanced combination therapies, as it's also been called. For example, the Victriva study is a combination of vedolizumab with and without upadacitinib, and then maintenance with vedolizumab alone in Crohn's disease to see if you can enhance the clinical remission rates in the beginning, and then maintenance is with vedolizumab, see if you can maintain it. I think this is intuitively an appealing approach because if you think of what we've done historically, often we'll try to get patients into remission with something like a steroid, but we might maintain with something altogether different. Of course, we know that JAK inhibitors are mostly safe, but we do have some concerns about immune inhibition in the long term. So this would be a very appealing approach. We're sort of using a JAK inhibitor as we currently use corticosteroids, but I would suspect that in the short term, JAK inhibitors probably have a much better safety profile. So this will progress over the coming years. We'll see what this yields.
This was a fantastic session, and I want to end this by asking you your thoughts on where the field is going. We talked about new MOA, combination of therapies, and I guess my first question to you is, do we need new MOAs, or do we need to understand how to better use what we already have?
I honestly think we need both of those things. And here's my thinking. Yes, we can get a lot more out of the drugs we have if we use them better. If we do treat to target, we are going to get better results. Maybe more importantly, and we haven't talked about this, is early use of effective therapy. We learned this in the profile study where very early treatment with combination therapy of anti-TNF and thiopurine within days or a few weeks of onset of Crohn's disease after diagnosis produced phenomenal efficacy. So we're not using effective drugs early enough. We could get a lot more out of them. But the other thing is, even then, we still have a lot of patients who are refractory to therapies. And the thing about combination therapy is right now, we're just combining things that we have. And all the things that we have, in one way or another, are really suppressing the immune system. So you can imagine that piling on immune suppression or altering the immune response is going to have its limitations, both in efficacy and maybe also might show safety signals. And there are a whole bunch of other very interesting mechanisms that are not being addressed potentially that are relevant to the pathogenesis of IBD. For example, the microbiome. We do not have an effective way to manipulate that now. For example, barrier function. We know that's aberrant in both Crohn's disease and ulcerative colitis. We don't have agents that address that. For example, T regulatory cells, which don't suppress the immune system, but just sort of bring it into equilibrium again. We're just starting to see drugs and approaches that come forward now that address that. Once we have those newer MOAs, combining them with things that are immune modulating will complement what we already have and might bring us to new heights of efficacy.
I agree. I think as the diseases we're treating are becoming more complex, we need different MOAs that probably intervene at different site of the pathogenesis. So maybe I can ask you, you reviewed with us some of the combination therapies that are up and coming. How do you combine therapies? How do you choose the right patient that you feel would benefit from combination of effective therapies? And what is your preferred combos? And, we talked a little bit about those today, but I was wondering, what do you do in your practice?
Right now, combination therapy in practice is a bit of a mess because it's opportunistic. It's rare that we would get a insurer for a patient to allow us to intentionally do combination therapy. So what we can do is someone has psoriasis and the rheumatologist is prescribing a JAK inhibitor or maybe an anti-IL-23, but we're prescribing the anti-TNF for their Crohn's, or maybe it's vedolizumab for their Crohn's or UC, and we're sort of getting at combination in that way. So that makes sense if you have two immune-mediated conditions and you're treating each one a little bit differently. That's one way to approach it. The other way in which we do it is obviously we have all got these patients who are absolutely refractory to monotherapies. They're out of options. Then sometimes we can put combinations together. This is not well studied. It's all opportunistic. You'll see case series. There isn't any compelling safety story there, which is interesting and good. But on the other hand, we don't have very robust efficacy data either. So it's kind of the Wild West. It's trial and error. You do, and you see for your patient in a treat to target fashion what works. So I can't say that I have some preferred combination, really.
All right, there's no secret sauce. So
I wish.
right. So we have to personalize to the patient needs and phenotype and prior exposure to therapy and what's available to us, what we can provide the patient, what therapies can be approved. So maybe what I would like to do is, as we wrap this up, say, while we're reviewing all these new MOAs or new trials, one thing that was very striking is that, like you pointed out, is that we are doing trials in a different way. Where do you see clinical trials going to be more kind of efficient and answer the questions that we're asking to ourselves in our practice? Is this drug the right drug for the patient? How would you design that trial?
It's a great question. And the one factor that you didn't incorporate in your question is what is compelling for regulators. In the United States, there's a strong preference for new molecular entities to be compared to placebo. We wish in the field that there would be allowance for comparison to active comparators, such as you saw in Galaxy. But even Galaxy, they incorporated placebo as well, right? So we would love to see the elimination of placebo. We'd love to see shorter studies. Why do we divide our studies into induction and maintenance? That's sort of an artificiality. Yes, we want early response and later response. We want to understand kind of the timelines of response, but that's not the real world either. Re-randomization of patients in response, we don't do that. We treat and we see if people respond or don't respond. We continue if they're getting better enough to allow continuation of treatment, we do it. So we probably don't need 1-year studies to determine if a drug is effective, probably even 6 months or 9 months. We can figure that out. And in fact, in other immune-mediated diseases like rheumatoid arthritis, both of these changes are permitted, but haven't yet been permitted in IBD.
Okay. Well, that was a wonderful conversation. Thank you so much for joining me today and sharing fascinating data and your thoughts on what's next. We reviewed new MOAs with very good safety. We reviewed combination therapies, and we reviewed new method of delivery of drugs that we're already familiar and trusting. So very exciting to be in the field of IBD. And thank you for making things more simpler for us into how we can approach these different therapy in our toolbox when we're seeing our patients. So thank you to everybody who joined us today. Please make sure to claim your CME credit by completing the PUS survey and evaluation form in the links below or by scanning the QR code that's shown on your screen.